All, this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show for today. So we are with the long weekend. I was actually planning to do a long marathon over this weekend, but I think it is a long weekend. People will be busy, and then some people are going to be busy with the Memorial Days activities as well. So we would move this marathon from this weekend to maybe some days in during the week or the next weekend. So that is one news. Second is that there is a lot of discussion about the vaccinated people shedding, to the point that somebody reached out to me and said that one of my patients, so some healthcare worker, reached out to me and said, one of my patients, a female patient, is asking that should that patient and her daughter, move away from her husband, daughter's father, because he has become vaccinated. And they were afraid that there would be irregularity in periods and such things. And this was not out of spite that why did the husband get vaccinated. This was out of concern. So that kind of message alarmed me, that we have discussed this every once in a while here and there in smaller chit chats, but we have not actually looked at it holistically together. So today, main concept I want to cover is the outcome of these spike proteins. Will they be shed from a person who is vaccinated or not shed? And if you wanted, I could also extend this discussion into, will a person who is vaccinated, will they shed a virus or not? So we can have that discussion. Again, I realize that someone who is not going to believe it, there are folks who are not going to take a vaccine, and for that they have certain set of concepts that they have been adhering to. And when I change those concepts, it's not necessary that they go along with me, and I'm not expecting it. But those who are cool beans, who understand that these mechanisms are important for us, I'm going to explain it, and hopefully that would work. So I'll have to block folks who are putting these kind of messages. So please stop, or I'll block you. All right. How is everyone doing? Let's do a very quick chit chat, and then we'll have this discussion again. So Anna Strange says, not first again. Jody says, question, can you please explain how the excess deaths in a time period is a valuable reporting of deaths from COVID? Thanks, I love statistics, although I find it difficult subject. So the excess death can be because of many reasons. At this time, given that a country is a hotspot, for example, India, or the whole world at this time is a hotspot as well. So excess death nowadays, so there is an expected level of deaths daily, monthly, yearly, and then there is extra. And those extra deaths can be for many reasons. There could be outbreaks, there could be pandemics, there could be floods, there could be other disasters. So here, nowadays, stable countries which do not have other disasters, when they have excess deaths, they are usually counted as COVID deaths. And especially in the kind of areas of outbreak, it is even more relevant. Okay, so Michelle says, yes, Dr. Queen, I wish that you would have that conversation. I have a friend who's living with an anti-vax. Her that mask makes him stay in this room because he's vaccinated. Yeah. So what I have seen, Michelle, is that it's not necessary that folks who are holding on to these beliefs will change their belief, but at least we should look at the science together. So I'm going to have that discussion. So let's start. There is a Claire Robinson says, crazy ideas, getting virus vicious spikes from vax. Where do they get these ideas? I think that, look, not everyone is medically detail-oriented or able to go to the right places to get those data points to be able to make up their mind. So sometimes people are just hearing from other friends, people they trust, and whatever is a rumor, that is what they accepted. So I think it is interesting to kind of talk about it. Dr. Amir Hashmi says COVID cases decrease in India last couple of days, but mucormycosis is occurring. So we've talked about how to manage mucormycosis. Ruby Zaret says, you made me laugh out loud in the grocery store. I enjoyed your discussion debunking theories so much. Thank you for making me laugh. You are very welcome. If you look at the commentary down there, compared to my older time, the commentary has become much more reasonable. In the older time discussions about vaccines, there was much more intense anti-vaccine folks reactions. Now that is less, but still most of the time the comments are, we will go with what a Nobel laureate says compared to what you said, how many Nobel prizes have you won, and so on. So that I feel that no single person's message is accepted by everyone. If that would have happened, there are many, many better personalities, even better than Nobel laureates. For example, there have been prophets, there have been leaders who are much, much more important and blessed and gifted than a Nobel laureate or me. And even sometimes their messages are not fully adhered to or listened by everyone. So who am I? So having said that, science does not change. If a Nobel laureate says wrong from a scientific point of view, then they're wrong. Having gotten Nobel once does not mean they can keep inventing new science and that would all be okay all the time. So that means I can be wrong as well. And I'm not even a Nobel laureate, so I can be wrong more, fine, but they can be wrong as well. So with this, let's start. So there is a question. Louis says, have you caught up on the protocol work yet? So no. So now I'm going to complain about myself. So I have been working on these things for the last few days. I've not been able to work on the protocol because of some of my own situation. My hand has been bothering me, and I talked with my doctor. I can't sleep well nowadays. My arms and my hand becomes numb. And so spoke with the doctor and I said, I think I need physiotherapy and I need imaging of my cervical area. And they talked about that I've been spending 2-3 hours daily presenting and then 3-4 hours planning and drawing. So they wanted me to go to physiotherapist to understand if my setup is ergonomically correct or not, and if I'm using my hand and arm correctly or not. So I have been going to doctors for the last week. Even today, before this lecture, I was at the physiotherapy. And so they are trying to correct my workspace and they're trying to get me back in shape. In addition to this, doctor has also started giving me some of the medicines to say, you know what, just relax a little bit so that you're not causing so much of trouble with your cervical spine. So because of some of these extra activities, I was not able to focus on the long hauling, and that I feel really bad about. So before this meeting, after coming back from physiotherapy, I was on a call with Dr. Pierre Corey, and he was I was making fun of him and he was making fun of me. When we speak, we have a good wavelength with each other. So he was saying, Mubin, what are you doing? Every day in our team meetings on FLCCC, they ask me that where is the long haul protocol? And I say, I'm working with the team to put it together. And he said, in my heart, I say, Mubin, where are you with the protocol? And then he was encouraging me to say, whatever it is, we'll put some disclaimers. Let's put it out there. We'll continue to polish it. So I promised him that this is a long weekend and I'm going to spend my tomorrow's day, which is an off day from teaching, and put that together. And he said that he's okay with that. So my apologies for this, but this is the truth that I'm putting in front of you. All right, so now let's start with this question of, will a vaccinated person shed? and I had not prepared it before, so I don't have pre-prepared diagrams. I'm going to go diagram by diagram and clear. Here is how we will talk about it. Number one, we'll look at various vaccines and their types. Number two, we'll see what do they produce. Number three, we'll see when they produce something, the result of the vaccine, how far, how wide can that go? And I'll talk about some of the myths and rumors as well that I'm hearing from people. So there are people who talk with me independently, and when I say this would not happen, they will say, you do not know, and this guy said it would happen, and so on. So I'll explain some of the counter thought patterns as well. So let's start. Let's say we start with um, messenger RNA vaccines. There are two vaccines in that group. One is the Moderna and the other one is Pfizer BioNTech, correct? These two vaccines, the way they work is the following. So they're given in deltoid. So what I would request you is to keep an eye on what will happen once a vaccine is given. Will we have spike proteins? Will those spike proteins in some significant way spill out in the blood? From there, will they be able to reach a person's respiratory system? From there, will they be enough in enough quantities to actually be shed from that person? This is what we have to keep an eye on. So here is the deltoid muscle. There we injected some vaccine. Now let's look at the first chance. So we go one point by point. First chance of, can this vaccine directly enter a blood vessel? Doesn't even enter a cell, just directly enters a blood vessel. And what would happen if that happens? So first point's answer, vaccine is made up of lipid nanoparticles. These lipid particles are very big. And I have done that discussion before as well. They're quite big. They cannot pass through the gaps in capillaries. Now, I would give you all of this, that how do you verify it? So please do me a favor, just look up for capillary gap junctions. Capillary gap junctions. This is not new science. This is just science. Capillary gap junctions are the spaces between the cells of a capillary from which things can come out or go in. If you go to the bigger vessels, so in the tissue, vessels become very small, they become microscopic, they become thin so that they can do exchange of nutrients and gases. And then as they come in, they were actually thick before, and as they leave, they become thick again. So the thick part of the blood vessel has a wall on it, and that wall would not allow anything to get in or get out. So the only chance for any material to get into the bloodstream is to either by a broken, ruptured, damaged blood vessel or by a natural process of entering through the gap junction. If something is broken, ruptured, and damaged, then anything can enter the blood vessel. But even then, as you know, when we get injured, we do not start getting so many of the things entering the blood vessel that we die from them. So here, first, healthy blood vessel capillary, this nanoparticle cannot enter a blood vessel. Can it connect with the cells of the blood vessel? So let's make one cell here which represents the endothelium. Endothelium is the cell of the blood vessel wall. So let's say it can enter an endothelium because it is made up of a lipid, and the function is to have the lipid enter a cell. So sure, if that is the case, it can actually enter an endothelial cell. So that is one possibility. Then it can also enter in the same place here. It can also enter local cells that are called fibroblasts. It can also enter a muscle cell over here. It can also be picked up, which is the most common outcome, is going to be picked up by macrophages, neutrophils, dendritic cells, or the local immune system cells. These are the cells that can pick it up. So the first thing, can it enter the blood? Very, very rare. So now I'll add this myth here that some people would say, well, what you don't know, doctor, is that the needle would go into the blood vessel and directly inject the vaccine in there. Look, we have decided for the deltoid muscle, for the reason that blood vessel is really deep, small, and next to the bone. It is not easy for even accidentally rupturing that blood vessel and adding the substance to it. And I have done a complete 1 hour discussion with the anatomy of this area to say why this is not easy. So we can say that, let's say out of 10 million, maybe in one case that would happen, but that is a rarity and an exception. And if in that case that happened, we can discuss that separately. So now, if it does not enter a blood vessel and enters a cell, endothelial cell, what is in it? Does it contain spike proteins? No. It contains the messenger RNA, because this is another common myth that, hey, vaccine contains the spike proteins which would just start spreading in the tissue and in the blood. That is not the case. The vaccine itself only contains the messenger RNA. That messenger RNA has to enter a cell to manufacture spike proteins. It cannot, outside of the cell, make spike proteins. So what would happen if there is a lipid nanoparticle that stays outside and it breaks outside for some reason? If that happens, the messenger RNA would come out of it, and that messenger RNA will be destroyed by RNases. RNases are enzymes that are present, and DNases are enzymes that are present in the tissues, which would hone in and kill this RNA. Even if they don't kill this RNA, even if they don't digest this RNA cannot produce any spike proteins, because it would need to be inside a cell to use the cell's machinery to produce spike protein. So then what would happen if RNases do not destroy it? The other possibility is for a phagocyte, that is macrophage, a dendritic cell, or other neutrophils, to pick it up and destroy it. So even if we say that this RNA can enter a blood vessel, it still is of no use, because it cannot produce a spike protein or a virus. So far, the concept of shedding is not applicable, because the particle and RNA has not gone anywhere. Okay, now let's address the second part of this. And that actually, let's address this as well, that imagine for some reason the blood vessel actually did get broken. So here is a blood vessel. It did become broken for some reason. Let's say the needle hit it, which is really not going to happen, but let's make this happen for our theory. So needle has hit the blood vessel has broken, and the lipid nanoparticle has entered here. Now these lipid nanoparticles are nothing that our blood cannot handle. Why not? We actually have bigger particles made up of lipids, which are called chylomicrons. CHYLO, I think, not ILO, chylomicrons. And these are the terms that I would request you to look up and read, because these are nothing that are unique science or only Mubin is talking about. Chylomicrons are the lipid particles that are formed when we eat food and we digest and absorb lipids. And these particles are the one that you do when you go to a doctor and you do your HDL or LDL or ILDL and VLDL type test for cholesterol. They are actually measuring these particles and their densities and their composition. So these are nothing new for our body. So if we have a chylomicron-like particle, a lipid nanoparticle, running around in a blood vessel, what would happen is at some point it would be picked up by some blood vessel cell, some endothelium. And then same thing would happen that I'm going to talk next. So a lipid nanoparticle that is picked up by cells here in the tissue of the deltoid muscle, or if it is picked up by an endothelium from a damaged blood vessel, let's see what happens next. So now let's bring this thing inside a cell. So inside the cell, lipid nanoparticle comes in and it is broken up. It is broken up and the RNA is released from it. So now it is inside the cell. This RNA, first thing, will not live here forever. And I am aware of people sending me studies that say this RNA is going to live here forever. It just is not going to be able to live there forever. And the reason is very simple. The reason is, please realize, our cell's function is the following. Whenever we are going to make something, let's assume a cell, endothelial cell, blood vessel cell, and that cell's job, endothelial cell, makes a lot of things. One of the job that we are all aware of is to make ACE1 enzyme and ACE2 enzyme. Why are we all aware of this? Because we're talking about SARS-CoV-2 for a year. So we know that on the cell surfaces, there are ACE2 enzyme. So remember that now ACE2, to make ACE2 enzyme, there will be some gene in the DNA that would have recipe to make ACE2 enzymes. So what happens is when we need it, we open that gene of the DNA in our nucleus, we copy the RNA from there and bring that RNA out. Then that RNA is fed into a machine called ribosome, which will make ACE enzyme. Those ACE enzymes will be collected together in a little pocket, and then finally that pocket goes to surface and enzymes are attached to the surface. Now this RNA that was here, this RNA is not going to stick here forever. Our cell cannot afford to keep collecting junk. This is like if you're eating food at home, then you are the fecal matter and the urine and the food particles and the packaging all stays within the home. How long can that happen? You can imagine that it won't be very long before the house becomes a trash. Similarly, our cell cannot afford for RNA to stay here forever. So what would it do? It would destroy this RNA within 1 day, 2 days, 3 days. This RNA that is gotten into our cell through a vaccine can also not live in the cell for more than 1, 2, or 3 days. So for 1, 2, 3 days, this RNA would continue to work with ribosomes and continue to make spike proteins. Good. So now we have reached a point where the alarm should go up that, okay, fine, now we have spike protein, and now we're going to think about shedding. Okay. First, let's clear this out, that this RNA would be eventually destroyed within 2 to 3 days. So there is no concept of making this RNA, making spike proteins forever. There is also no concept of this RNA becoming introduced in the DNA like a retrovirus. Those scientists, they may be Nobel laureates or they may be whatever. Those scientists that say that this RNA will become part of the DNA, they use a crutch, a scientific crutch, which is incorrect. And let me explain that crutch to you. They say that there is an HIV RNA present in the vaccine or present in the virus. Why do they say that? The reason for saying that is that HIV virus is a retrovirus. A retro means the other way around, inverted. Retrovirus because it has an RNA that can be converted into a DNA, and that DNA has a possibility of incorporation into our DNA. So they use that crutch to say, hey, man, you know what is going to happen? Your vaccine RNA is going to become part of your DNA through the retrovirus enzymes. But there is no retrovirus in this RNA. And I'll explain, there is another answer they give to this. Also, the actual virus, the SARS-CoV-2 does not have retrovirus. This is why that study, which I showed you a few days ago, where we talked about some people in January of last year saying that we have HIV virus's RNA present in the spike protein, which they then retracted. And many people commented to me that they retracted it out of fear. No, even if they had retracted it out of fear, there are going to be much more researchers and doctors sitting in independent countries where they may not have that kind of fear, and they would be able to talk about it. None of the other researchers were able to find those in there, and we actually saw that those pieces were present in other coronaviruses, were present in other pathogens as well, and present in other species as well. So that was not a new thing, and that was not an HIV virus. Why are they continue to connect this thing with HIV because they want to use the concept of retrovirus to tell you that this RNA would become DNA and become incorporated in your cell and live there forever. And that is not possible. So continuing with this one, that we have an RNA over here, and this is the case with the virus, or this is the case with the vaccine. There is one more concept that I saw by one of the other such clever people. So I don't take them to be fools. I just take them that they are pushing incorrect science. So one of the more cunning one in these folks have said, you don't need the vaccine to have retrovirus in it, or retrovirus enzyme in it, or RNA in it. You also don't need the virus to have the HIV enzyme in it. Our cells always have retrovirus enzymes. So whatever will happen, this RNA is going to become incorporated. And I think some of you would recognize who said that. Problem is this. If we take that on this face value, that our cells have retroviruses in them, or retrovirus enzymes in them, or retrovirus RNA in them, then all of our RNA, which is produced for normal cell functions, will continue to become converted into DNA and getting inserted into the nucleus. And that nucleus is going to die within a day or two, or it will become cancerous. So it cannot survive. So that is also wrong. So first, this RNA will be destroyed in 2 to 3 days. Now the product of the RNA, this RNA would be fed into a ribosome. That ribosome will make a spike protein. And we have done this discussion many times, that this spike protein will be brought into an endosome or proteasome, where it would be destroyed and then it would be presented on the surface. And this is why there is no concept of this spike protein starting from the deltoid muscle, going all the way to the throat and then being shedding out. Even in the deltoid muscle, you would not see spike protein. You would see pieces of spike proteins shown on the cells here, both muscle cells and immune system cells. Immune system cells which are presenting this, for example, macrophages or dendritic cell, will go from here to the local lymph nodes and present those antigens, pieces of spike protein, to the T cells and B cells in the lymph nodes. And that is as far as a broken spike protein will travel. And I have said this many times, which it is interesting that folks then put my own message back to me. So I have said many times, the only way to get the spike protein out of this cell is to break the cell. So what does that mean? We have to now break one more concept here. That is another concept propagated by another such doctors or scientists, not very clever one though. And her point of view is that this RNA is going to become incorporated into the DNA, which would then start producing spike proteins forever, and our cell would generally present a spike protein on its surface like this. So that is impossible. The reason for that to be impossible is that here are the things that need to be done for a cell to start producing a foreign antigen on its surface as a regular self. Here are the things that need to be done. Number one, this RNA has to become converted into DNA. Then that DNA has to become attached to our RNA. And I've talked about CRISPR technologies before. It would be very difficult to add a piece of DNA into our normal DNA. Even if we try to do that, then we have a reviewing system within our cell to make sure that that DNA does not break down. And please look it up. Don't trust me. I keep getting messages from people saying, why should we trust you? Don't trust me. But look it up. Look up CRISPR technologies. Look up the difficulties and ease of the CRISPR technology and try to understand, is it going to be this easy to modify a DNA? So, number one, incorporating that in the DNA is going to be very difficult. But let's give it that our DNA will have a piece of DNA added to it, which will help make spike proteins. Second part, that is needed is the cell now has to understand to produce this protein and then put it on itself outside. For that, please realize our cells that we have thyroid cells, we have skin cells, we have mucus cells, we have eye cells, we have GIT cells, we have muscle cells, bone cells, immune cells, respiratory system cells, pancreas cells, liver cells, kidney cells, all cells, right? Why are they different? They all are different because they have decided to open certain genes that produce certain proteins which would make them do a certain function. Kidney cell would do kidney function because a certain set of pages in the book are open in that nucleus. Here we are saying, imagine this is my deltoid muscle. Here we are saying that not only you would be able to make a spike protein by incorporating this DNA in you, then you would be able to make those spike protein by opening that gene and continuously expressing it. To express a gene, what needs to be done is, if this is a piece of DNA, in the beginning of the gene, so let's say this is a gene. In the beginning of a gene, there is a thing called promoter boxes. Promoter, I do not know the spelling, ER or OR, promoter boxes. There are, or Tata boxes. So there are promoter boxes which allow various enzymes to come and stand there. And then those enzymes would order this area of the gene to open up, and then messenger RNA to be built from here. So what we would need is that this spike protein's RNA that got translated into DNA is now needed to be flanked by a promoter box, which would allow certain enzymes to come and attach here to open this gene and express this. So you have to provide a promoter box and the enzyme that would work with that promoter box. The problem is that for this enzyme to work with the promoter box, there has to be a signal to it to say, please go and work. For example, thyroid gland. The thyroid stimulating hormone from pituitary, when that is released, that goes to our thyroid cell and says, please bring the enzyme to the thyroid making promoter box gene so we can open that gene and we can make messenger RNA, which will help make thyroid hormone, which will help secrete thyroid enzyme or hormone. We do not have any promoter boxes automatically created. We do not have any such new enzymes created in the cell. And I know that folks who don't want to agree, they would do two things. Number one, they'll curse me and say, you suck. And secondly, they'll say, this all will be created. Whatever is necessary will be done. So in such cases, I have no solution for you. Then that is between you and your body and your discussions with your doctors and your community. I have no business there. And I have no influence there, and neither do I want to have any influence. So back here, this is not possible. So go back here. Can this become part of the DNA? No. So can this cell present the spike protein on its surface forever? No. Even if we give the cell this, remember we said, fine, we'll give this mechanism that RNA would become reversed and become part of the DNA. We give it. Let's give one more. Let's say promoter box will be added as well for some magic. By some magic, enzyme systems will be added as well to open the promoter box. Signaling system would be added as well to send the enzyme system to the promoter box. The whole shebang will be made available. Do you know what is needed next? What is needed next is that this DNA, when this opens up, this DNA would have to create a messenger RNA. That messenger RNA would go out of the nucleus into the cell. The cytoplasm would take that messenger RNA, would make a protein from it. And in this case, this is the spike protein. And do you know what is needed next? We have to teach this cell to not destroy it, but instead present it on the surface. And cells do not present everything on the surface. They present things on the surface as an antigen, with MHC1 or 2, they don't present them on the surface as clusters of functional proteins without those cells being of that type. So even when the spike protein is created, look how many provisions we have given. The fate of this spike protein is going to be destruction in an endosome and presentation as an antigen. And the fate of the cell will be destruction by the immune system. This person will die within a few days, whose DNA just got changed. Not even months or years or 2 years later or 6 months later. When these cunning folks say that, what they're doing is that they are making you believe it now and then putting the results in future, which would never occur. So you have to now take it on that face value that maybe in 6 months people would die. This spike protein will be destroyed immediately and presented on the cell's face with MHC1 or MHC2, and the immune system would react and this cell will be killed. So if this happens with all these provisions, even then the person will die, they would not survive. So the concept of shedding from this person is not going to be there. They'll just be dead. So this is not going to happen. And this is not happening with the virus, and this is not happening with the vaccine. This is not happening with the variants. So let's go back. Now, what are we left with? The only way to bring spike proteins out. Now we're still talking about the vaccine, have messenger RNA, spike proteins are produced, and we are trying to bring it out. So I was telling that there was a cool bean who sent to me a message saying, or commented that surely you know that there will be these half-built spike proteins. I would add to his message to say fully built spike proteins that are not yet destroyed, which would get out when the cell will be dead. That's true. Yes. When this cell will be killed, and not every cell will be killed, but if this cell will be killed, and if there are proteins present in here, those are going to leak out. Correct. Now, next part, the cool beans who have been with me, they know this, that this would get out and they know what would happen next. What would happen next is, again, this is a deltoid muscle. It is happening here. This whole drama is going on here. We have a macrophage here. We have a neutrophil here. We have a dendritic cell here. They are going to pick it up and eat it up. They are going to destroy it. Now it's going to be presented on MHC2 instead of MHC1. These cells would pick it up from here and go to the lymph node and say, hey, T cells and B cells, look what we found. And when they are presenting it on MHC2, once again, they have destroyed this thing and presented it. That is the majority of what would happen to this. Let's give one more provision to the folks who said, no, I still want the vaccine to shed. So let's create one more area. And let's say some of this protein that will leak out. So look how much reduction in chances is occurring. That now we need the vaccine to produce messenger RNA, then that to produce spike protein, then that spike protein to somehow come out of the cell by destruction of the cell itself, and then escape the macrophages, neutrophils, dendritic cells. And these cells are not that easy to escape. But let's give the next step. Let's say this would escape, and now this spike protein has to enter blood. Why? It has to enter the blood system to be able to somehow reach the throat and then come out of the blood system. Look, we're thinking about this thing and we are giving so many privileges to be able to get shed. And I hope that if you just listen to this from an open mind, you would think that this is not going to happen. But let's continue. Let's say the first challenge for the spike protein is now to enter the blood vessel. And the challenge is the same that was with the lipid nanoparticle, that how does a spike protein enter inside the blood vessel by passing through the gap junctions? it is not easy for the spike protein to pass through the gap junctions because spike protein is very big. And I'll give you an example to kind of do your research. Look up albumin. Albumin is the smallest protein inside the blood vessels. It is the smallest protein present inside the blood vessel, and even albumin cannot come out of the blood vessel system. And those people whose, let's say, renal system is incorrect, or whose protein liver system is incorrect, or whose blood vessel they have vasculitis, they know if the albumin comes out, what happens? They get edema, they get severe situations that can be life-threatening. Here we are saying that even a thing like albumin cannot come out, but a big spike protein sitting here would somehow be able to enter this blood. But let's do that too. It has entered the blood. Now we are hoping, so let's cheer for this spike protein, that it has entered the bloodstream, and let's say, come on, go to the throat and become expressed here and become part of the saliva, so when the person talks or laughs, it should be shed. But do you know what is going to happen next? We know that spike protein likes to bind with ACE2. And this is also it's a fact, but it is also used as a conspiracy theory to say, you know what, just the spike protein itself would cause vasculitis. So let's look at that. Let's say here is an endothelial cell of the blood vessel. This spike protein was able to enter the system, blood system. Do you know what it's going to do? It's going to just bind with the ACE2. It's not going to go to the throat yet. It has entered the blood system here by some magic, but we have allowed it to enter there. Now it is going to attach to ACE2. That is where it belongs. And ACE2, when it attaches there, we know what would happen next to that. We have done that discussion. Either it can be taken up, or it would just be there until it is destroyed by the local inflammatory cells, because it causes inflammation. Now, do we have sufficient spike proteins to cause systematic inflammation? Unfortunately, no. And why do I say unfortunately? Because there are folks who want me to say there will be so much spike protein that the body would be destroyed. No, this is a spike protein, tiny amount produced. So now I can convert my unfortunate to fortunately no. It's not enough. So it would go into some blood vessels. It would probably cause some inflammation there as well, which would be gone within few hours, and you would probably just have some local reaction and done. When will the body become responsive to a huge reaction? If the antibodies are produced, not spike proteins are produced like this. Then why does virus do it? Virus gets into a cell, replicates there, becomes millions, comes out, gets into the next cell, replicates there, becomes millions, comes out. This hand of the virus, even if it enters the cell through the ACE2, it doesn't have the rest of the enzymatic system with it. What would it replicate? Nothing. It doesn't even have an RNA in it. There is no genetic thing. It's just a tiny protein. So if the cell would take it up, cell is going to destroy it, and it's going to present that as on MHC1. If it just binds with the ACE2, it's going to cause some local one cell inflammation, which our system is not going to even bother. But will this spike protein get a chance to go all the way to the throat and cause inflammation and shedding? Not inflammation, shedding. Not going to happen. But let's give it this as well, that it would actually go to the throat. So let's say this spike protein somehow escaped all the ACE2 jungle on the way, and it went to our area of the throat. Now, what is the challenge for this spike protein? The next challenge is that in this area, it has to now come out of the blood vessel. Again, it has to save itself from binding to an ACE2 incorrectly. It has to come out intact, out in the tissue, and then become part of saliva. And then from there, it has to be shed. when the person laughs or does those. Would that happen? When it is coming out, the tissue, the mucosal tissue cells, epithelial cells over here, did they have a lot of ACE2? Would they not catch it, or will it not catch them and attach to their epithelial cells ACE2s and get absorbed there with the epithelial cells and be destroyed? So even if we then give it this provision that, man, even epithelial cells are not going to say you anything. You are a privileged, very important person, guest, do what you want. Then this spike has to sit in a droplet and go to another person. And it has to go there in enough quantities to cause issues. That's not going to happen. Now, I want to add one more route to it. And I know people are not going to bring it to me because they're not going to think about it, but I'll think it with you. Another route from this area to bring to throat will be not through the blood vascular system, but through the lymphatic system. So let's say there are big lymph channels that are from here going to the lymph node, and from the lymph node, eventually the lymph goes into blood. So folks can say, more medically trained folks who can think about the this lymphatic system as well, they would say, you know what, Mubin, these spike proteins should simply just enter the lymph, and from the lymph to the lymph node and then to the blood, and now voila, they're in blood. They don't need to cross the gap junctions. Two answers for them. Number one, even if they enter the blood this way, they still have to exit the blood vessel and pass through the epithelial system to become part of the mucus, the droplets, and then shed. One problem, which in itself is sufficient to say it's impossible. The second problem, when this spike is actually going through the lymph, you know what is the function of the lymph. Those who know lymph, you know that the function of the lymph is to pass through the lymph nodes, where there are cells that are sitting to catch things that are coming in and filter them out. The basic function, other than the immunological function, the function of the lymph is to allow bigger broken pieces of the debris in a tissue which cannot enter blood vessels to go through the lymph into the blood system, but be filtered out on the way. So this is a blood cleaning system as well, in addition to the immune system. So when this spike protein is going to reach the lymph node, it's going to be cleaned out over here. It's going to be picked up by macrophages, and it would once again be presented to the lymph node and voila, destroyed. It's not going to come in here. If our body was that poorly built, that lymph with all the debris in it and with all the toxins in it that it collects from some tissue, which may be burnt, which may be damaged, which may be infected, and that just simply end up in our blood vessels, then our heart will be always in trouble, because it would get toxins, it would get bacteria, it would get viruses, and it would just, every other day our heart will be in trouble. Our blood vessels will be in trouble. That doesn't happen because lymphatics have a huge sophisticated filtering mechanism. So now we have to deceive that too. So imagine what we are saying is, please give me a person's system where everything fails so that they can shed. But I can guarantee you that if all of these mechanisms fail, that person is not going to be living with you and talking with you and breathing near you or eating with you. They would be in a hospital or dead if these mechanisms have all failed to bring a spike protein to the throat. So this is with the messenger RNA. Now let's look at, can they shed? So here, I hope that you've seen all the mechanisms. You can do your own research to see where can these mechanisms fault and allow the spike protein to arrive at the. So now I'm going to go to the second vaccine. I'm not going to repeat all the mechanism, but just going to repeat vaccines function. So now let's say instead of messenger RNA vaccine, now we have an adenovirus-based vaccine. So adenovirus-based vaccine means Johnson & Johnson vaccine. That would mean AstraZeneca vaccine. That would mean Sputnik 4 or Sputnik 5, whatever is the right pronunciation, but Sputnik 5, whatever those vaccines are. These vaccines have a adenovirus. This adenovirus has a DNA, cDNA. So here you would say, voila, I got it. Now we got Mubin. There is a DNA already in there for spike protein. This DNA is trouble. And I'll explain more trouble. Again, those who have been here with me for some time, they know what would happen next. What would happen next is this adenovirus has its own spike proteins. And these are different kind of spike proteins, but they are spikes. They would attach to our cell. They would be the virus will be phagocytosed, and then it would be brought in a phagosome. This phagosome will be then connected with a lysosome, and then it would be destroyed. No immunologist, virologist, Mubin, Nobel laureate, book so far can change these mechanisms. These are the mechanisms that are known to operate in our cells for centuries. They're not new mechanisms. They're known and they're very well established mechanisms. These are like 2 + 2 = 4. So now these are facts of our cells. Now the lysosome here, which has gotten the acids, will connect with this, and they start kind of destroying and damaging this adenovirus. This adenovirus would then broken pieces or half broken adenovirus. What is half broken? The spikes are removed. So limbs are detached from it, and the body with the DNA is in it. That is brought into the nucleus by kinesin proteins. Again, please don't go with my spellings. Kinesin proteins. I'm a doctor. I'm bad with spellings. That's why I'd like to write like this, kinesin, done, because I don't know the spellings. So kinesin proteins would haul this broken, half, partially dissolved adenovirus to a nucleopore. And then this adenovirus is going to inject the DNA into our nucleus. DNA into our nucleus. And there is a study, which I have to go over with cool beans, which is showing that it is possible that this adenovirus may survive in a cell for a longer period of time. Now, there is a reason if that can be done. There is a way that can be done. That could be done if inside the cell is the replication enzyme present for this. What do I mean by that? This adenovirus, which is brought in through a vaccine, it is specialized adenovirus that the replication enzyme has been removed from it. So it cannot divide in numbers. It cannot increase in numbers. Whatever number we injected are the numbers that we got. Now, for it to replicate, it will have to, from our own cell, figure out an enzyme that can help it replicate. And maybe, let's say, if I had some adenovirus enzyme virus in me sometimes in the past, this is just a theoretical possibility I'm talking, and that adenovirus's enzyme is sitting in the cell, which is really difficult. But let's try to make it happen. Let's say I was just infected by some adenovirus, cold virus. Then I got vaccine as well. And those enzymes were present freshly in my cells. And this adenovirus came into. Then this adenovirus may get a chance to use those enzymes to increase in number. This is a rare possibility. There is a study in which they said we are observing this can happen, and it can happen even for a year. So we have to discuss that study to see what was their study setup. Was that in vitro? Was that in vivo? Was that in vaccinated? Was that in animal? Was that in human? So I'll have that discussion. But there is a study like this. Now, this adenovirus is here. This adenovirus is going to release the DNA into our nucleus. Even then, we are not worried. This DNA is not going to become incorporated into our DNA. This DNA will be used to make messenger RNA. That messenger RNA would come out. That messenger RNA would help do all the rest to make spike proteins, which we know. Finally, this DNA would be destroyed as well. And I'll give you a simple example. If you say, no, I don't want to agree with you that this will be destroyed. We get cold many times in our life. Many times that cold is actually by adenoviruses. If every time an adenovirus comes into our cell and injects the DNA into our DNA, which is actually replication competent virus, that is not a replication deficient virus like a vaccine, it is competent, it can replicate. Even then, we do not allow its DNA to become part of our DNA, we destroy it. Otherwise, our cells would once again become some Frankenstein DNA graveyard or DNA place and make adenoviruses of their own and then just be adenovirus factories forever until they kill us very quickly. That doesn't happen. We get common colds all the time. This is the same thing. It doesn't have the replication in it. So it is actually limited, constrained, and the DNA it produces would be destroyed. If this DNA will not be destroyed, then the other adenovirus DNAs from the replication competent infections will not be destroyed, and we will then be destroyed. So then this messenger RNA comes in, it makes the spike protein would go out, and now all the rest that we talked. Will this person shed? No. Then let's go to the third vaccine type, which is inactivated virus. So in case of inactivated virus, and somebody sent me this message yesterday saying, don't you know that the polio virus can cause polio? This is why sometimes knowing less or not doing your own research and just to get back at me saying something that somebody else said I don't have enough time to have debates with people in comments. So I just ignore them. Inactivated viruses, or let me change my statement, attenuated viruses. Attenuated virus or a weakened virus, attenuated, once again, spelling challenge, or a weakened virus is a virus that is partially weak, partially treated to not be as aggressive. When that virus is injected in our body, we run a risk of that virus becoming active or giving rise to a daughter that is active and causing infection. That has happened with polio and other. Normally, what they say is that even if the infection occurs and the shedding occurs, that can cause shedding. Usually that is so low level that it is of negligible outcome, but it is possible. So they used that example to say, don't you know that this happens with the vaccine? So why would it not happen here? Well, the problem is, this is why we do not use attenuated viruses for SARS-CoV-2. That God forbid if we inject that and then it becomes active and makes a virus daughter that becomes competent and then it starts replicating, we have just injected the infection to the person. That person would shed. That person would have a problem. We do not want to do that. Remember here in US, there was a person who tried to make his own vaccine at home and ended up in ICU with the COVID. This is the kind of experiments he was doing. So not a good thing. And we do not have attenuated SARS-CoV-2 viruses. We have inactivated viruses. And I am actually afraid of inactivated vaccines as well. And I'll tell you why. But let me explain what are inactivated. So there are Chinese inactivated vaccines. There are India's inactivated virus vaccines as well. I actually like them from a mechanism point of view. I am just afraid of them, that if there is one particle sitting somewhere that may still be active, it may start an infection, may not. So what is an inactivated vaccine? We have talked about that as well. Here is the virus. It is a copy. For example, in Indian case, the inactivated virus, original virus is from Pune. That virus is taken, it is cultivated, increased in number in the labs, and then it is destroyed, not the whole virus, but the RNA of the virus, the genetic brain of the virus, is destroyed by chemical substances, so it cannot actually function. So you can say it is a dead body of a virus with the broken brain in it. And that is what we inject in the person. Now, can this lead to shedding? Again, if we have a good vaccine, then no, because this is not going to increase in number. Can this cause spike protein production? Yes, instead of making spike proteins with DNA in case of adenovirus or messenger RNA in case of messenger RNA vaccines, this would have its own spike proteins attached to it. And our body's cells are going to detach them, take it inside, detach them, and present them and do everything we saw. Will that person shed? No. Can this be a little more dangerous? It makes me nervous. That may be my ignorance, that I do not know how sophisticated is the mechanism to kill it. So if I say in that pile of the vaccine, if there are 10 or 15 that are alive, what would happen? But anyways, these are very good vaccines because they create a broad spectrum of antibody production as well. So then the remaining mechanism is the same as I just discussed. Now let's go to the next one, the sub-protein vaccines like Novavax. So here is how is Novavax formed? What they do is they have these spike proteins produced in separate cells in lab. So there is no messenger RNA, there is no DNA, there is spike protein produced. And then those spike proteins are fixed, just like edible arrangements. In edible arrangements, you have a arrangement and there are little sticks. On those sticks are fruits and stuff. Imagine there is a small particle here, and on that particle, they stick the spike proteins. So to our body, it looks like the virus. And with the body, it interacts with these spike proteins. If people are actually afraid of spike proteins, then this is the vaccine to be more afraid of, because this is nothing but spike proteins injected directly in the deltoid muscle. And now, can this create shedding? So once again, the problem is it will not reason. So all those remaining mechanisms and barriers, I don't want to repeat. But important thing is, it doesn't have anything to produce more spike proteins. One. Number two, the spike proteins that it has would be very quickly picked up by the macrophages, this particle, macrophages or the dendritic cells or B cells or neutrophils or even normal cells, but mostly this would be picked up by antigen presenting cells. When they would pick it up, they would destroy this whole particle, they would destroy the spike proteins, they would make small pieces of them and make them available on the surface. And that is how immune system would become trained. Can these spike proteins shed? All the previous rules that we talked are the ones that would be applicable. So all the vaccine types, none of them is going to be able to reach a point to allow so much of shedding. Let's say all of this mechanism set fails. I would not be able to understand what kind of a person will have all these mechanisms failed but still be a live person. But even if you give it that all these mechanisms have failed, the person still has some spike proteins. How many spike proteins are going to be here? It's not going to shed anything that would cause any damage to anyone nearby. Nothing at all, zero. And one final point is, can a vaccinated person shed the virus? So now not talking about spike protein, virus. The thing is, the more I discuss this with folks and I say this will not happen, they would just keep taking another position. Eventually they want to say that I am going to believe that person is going to shed. I can't do anything about that. But can a person shed a virus? A person who is vaccinated or a person who is not vaccinated, a person who is not vaccinated and has never been infected, or a person who is not vaccinated and has been infected in the past. They are all equal in terms of they can get infected. Can they send the infection back? Can they shed it? That shedding can occur depending upon how much IgA is present in their mucosa. So, for example, vaccinated or infected person may have IgA present in the mucosa, and that would bind with the virus. And even if it is shed back, it would be very low quantity. The quantity that came in is going back, and it would have proteins attached to it. So it would kind of be useless thing. That virus would have less chance of entering the cells because it is coated with these proteins around this, vaccinated or infected. And so that virus would have a difficulty to create new viruses to create enough load that the person sheds. So some studies with the vaccine have said that reduction in shedding is, let's say, 74% less. I still am doubtful about the studies to see exact numbers, because how do you tell if a person is shedding or not? Are you going to stand next to them with some sort of a capturing thing to say, as soon as you're going to breathe, I'm going to pick up the breath and calculate how many viruses are in there? That is not done yet. But infected person or vaccinated person would shed less compared to a person who is not vaccinated and who is not infected and has not, or not even infected asymptomatically and cured, meaning they are brand new for the virus. They would shed more because the virus would cause infection in them. So this is the discussion for today. I hope it makes sense. I realize that to some folks it will not, but it was important for me to bring it out and discuss it to see that will a vaccinated person shed or not. I'm going to do this now. Because it is a long topic again, I'm going to hang up and come back for a chit chat, and we'll do some questions as well. Let me see if there are any relevant questions here. Michelle says protein spikes of the or on the virus, protein spikes on the virus that attaches to DNA, possibly changing RNA. Not even, it doesn't even, there's not even a mechanism like this. Protein spikes, if I just quickly share my board as well to Michelle, kind of trying to understand so what you're saying is virus. So here is the virus. It has spikes on it, right? So you're saying that these spikes somehow attached to a DNA. So here is a cell. Cell has a nucleus. Nucleus has DNA. So this thing has to reach all the way inside the DNA to be able to do what you're saying, attach to DNA and possibly changing the RNA. The problem is the cell, this cannot reach a DNA. And if a virus has reached the DNA, it has actually destroyed the cell. Cell is gone. If cell is so open that the virus just entered all the way to the DNA. Not SARS-CoV-2. There are other viruses like retroviruses that have complicated mechanisms to reach. This thing, poor thing, doesn't have those mechanisms. It will have to attach with ACE2 and then get in. And when it gets in, it would have to stay outside and make a RNA of its own. It contains RNA and then do the rest of the thing. So this mechanism is impossible. So let's say Mr. J says here, can environment, race, or ethnicity affect how it mutates? Okay, so hear me out. The only possibility of it mutating in someone more than others, and this is an important statement I'm making, and you can verify it whichever way you like. Here is the thing. When the virus arrives in anybody who's infected, vaccinated, previously infected, uninfected, anti-vaxxer, pro-vaxxer, whoever you are, when the virus would infect you, I'm using you carefully. I don't mean that you should be infected. I usually use myself, but I'm trying to personalize this message. If you get infected, inside of your body, there will be many daughters created which will be similar to original, which would be more lethal than original, which will be less lethal than original. And they would be shed as well. And if there are more lethal, they would kill the person. If they're less lethal, they can shed out. If they're the same as well, they can shed out. So what is the possibility for virus to shed more? Mutations are going to be everywhere, because if you look at nextstrain.com.org, nextstrain.org, and this was a very interesting one. Somebody was really becoming annoying and was insisting that somehow these vaccines are going to cause this. And I said, tell me, why did we have all the variants before? And these variants didn't start with the vaccines. Here are the variants that started in January and then February and onwards. And their answer was, they're not important. That's not a good answer, because they all have mutations, and these are important mutations. And here they are. And I read this for cool beans all the time. I'm going to read it for newer folks as well. This is nextstrain.org, not me. They're saying there are hundreds of thousands of complete SARS-CoV-2 genomes available, and this number increases every day. This visualization can only handle 3,000 genomes. Genomes here are mutation. In a single view for performance and legibility reason. Because of this, we subsample available genome data for the analytical views. That he's saying hundreds, and he, she, it, whoever they are, hundreds and thousands of genomes, and we can only handle 3,000, and even from that we are subsampling. And if I hover my mouse over, so let's say this one, date, this is 4/10. So April 10, 2020, we had no concept of vaccines at that time. And look at the no nucleotide mutation, 6 divergences. And if you just keep going, you would see some, for example, here, nucleotide mutation divergence 7, and so on. Here is a nucleotide mutation. There is a nucleotide mutations. This is branch leading to USA, I believe, KYCDC. So look at the nucleotide mutations, 1, 2, 3, 4, 5, 6, 7, 8. Then amino acid mutations, 1 in here, the second, another, and another. So 8, 9, 10, 11. 11 kind of mutations present in it. The clade is 20B. We have never heard of it. And it has 11 mutations in it. And there are so many other. So mutations were occurring. This is not a right way to look at it, to say they are not important. Mutations would occur. They would occur in people who are vaccinated. They would occur in people who are not vaccinated, because our bodies are being used at a mass level by this bacteria, this virus. We should pray and hope that it does not develop a more lethal and more contagious form that becomes SARS-CoV-3. Okay, so so now, Mr. J, a person who is, I deliberately don't want to say this because it kind of puts some people not in the best light. But for example, if I am immunocompromised because I'm taking steroids, or I have an organ graft, or I have HIV, or I have some other reason for immunocompromised genetic problems, for me, the virus may have a more liberal time to mutate and shed a lot of kinds of it outside. So that is possible. Similarly, somebody who is weaker because of nutritional status, somebody who's weaker because of diabetic status, somebody who's weaker in other ways can allow the virus to replicate more. So there are many ways that these things can be. Vitamin D levels are important. Our immune system's suboptimal response can be important. So yes, there are ways that this can become a problem. Dr. Amir Hashmi says, Sputnik 5 vaccine efficacy in variants. I have not seen that data. Giladoni says, Giladoni, I saw you after a long time. All viruses continue to mutate all the time. Yes, very correct. So how about we do this? Let me hang up. This is already 1 hour and 18 minutes. And we do one more chit chat where we just do the chit chat as we do daily and then we stop. So while leaving, please like, subscribe, and share. When I have my guests with me, I do not say that just out of respect for them. So please like, subscribe, and share. And if you wanted to support this work, there are some links in the description that you can use to support this work. Thank you very much. And those for whom it is too late, have a nice weekend. Tell me this, should I take Monday off? This is a long weekend here in the US and Monday is off. If you're okay, I can take it off. If you want me to be here, then we'll have a discussion on Monday. And we'll do the long discussion maybe in the coming week or the next Monday. So I'm going to come back up once more. Thank you very much.